Hello students, I hope you all are doing well. So as of now we have finished the fertilization event. So the last concept in sexual reproduction in angiosperms are post fertilization events. So in this class we are going to study some post fertilization structures and events. So the post fertilization events are the formation of endosperm. Already the PEN is formed, primary endosperm nucleus, as we have studied in the previous class. That is, after the fertilization, what happens when the two male nucleus or gametes enter into the embryo sac through the micropyle end. Okay, they get discharged in the synergids, right? So from the synergids, one male gamete fuses with egg to form what? Zygote. Okay, and the ploidy of the zygote is 2n because our male gamete is n and egg cell is n, so formed zygote is 2n. So another male gamete, what happens? They go and fuse with polar nuclei or secondary nucleus. So the result of this triple fusion is primary endosperm nucleus, which is 3n in nature. Okay, so what happens? From the zygote, the embryo is formed. That is one of the post fertilization event. In the same way, from the PEN, endosperm is formed. And the rest post fertilization events are ovule structure. We know the ovule get converted into seed, or it get formed into seed, and ovary formed into fruit. Okay, yes. So we'll start with the endosperm development first. One by one we'll study. Next is embryo, then seed, then fruit. Okay, so here are some examples like how the endosperms are formed. So there are a few different types in your textbook they had given based on the endosperm development. The first endosperm, sometimes it is called as free nuclear endosperm, then cellular, then halobial it is called as. Okay, so what happens when PEN, this is primary endosperm nucleus, undergo repeated mitotic division where they cannot divide cytoplasm. So hence it is looks like free nucleus which is floating in a single cytoplasm. Hence it is called as free nuclear endosperm. So the best example for this free nuclear endosperms are our coconut water. Okay, so next is after repeated nuclear division, what happens? There will be cytokinesis. The cytoplasm divides and cell wall is formed around them, around all the nucleus. So cell wall formation takes place. Then the formed endosperms are called as cellular endosperm. Okay, so same in coconut fruit only. The water is represented as the free nuclear endosperm, as well as the white kernel where you see there. Okay, white kernel, the white part, the thick white part is nothing but cellular endosperm. So this is how the beginning of endosperm development takes place. So next is embryo development. Okay, in embryo development what happens? So in this diagram you can see after the fusion or fertilization, so here we got PEN, that is primary endosperm nucleus, which is 3n, and there are our antipodal cells as well as the two synergids get degenerated, and the remaining thing is the fused one, that is zygote, egg and male gamete, which is 2n. Okay, that zygote, what happens? They form, okay, they develop into embryo. So in dicot especially, so in dicot plants, these are the different stages of embryo development. What happens? First, there is a single zygote. In the diagram you can see there, single zygote. Yes, so this is the single zygote. So next what happened? This zygote divided into two cells, and the upper cells, okay, upper cell doesn't get divided, and the lower single cell, they divide into several cells by undergoing mitotic division. Such a kind of embryo we call it as globular embryo. So in behind you can see there is a globe like structure. Next, heart shaped embryo is formed. Next, what happen? Our matured embryo, where it consists of radicle, plumule and cotyledons. We know already what is radicle and plumule. Okay. Yes. So what is radicle and plumule? Already we know what is radicle. Radicle is the future root. Plumule is the future shoot. Yes, in first year we have studied in morphology of angiosperms, we have studied root will arise from the radical part, shoot will arise from the plumule part. Okay, so this is the difference between, this is the comparison of the embryos of dicot as well as monocot. You can see here, okay, the axis which is below the cotyledons, okay, the structure, cotyledon structure, we call it as hypocotyle, and these two are the cotyledons. This is the plumule where the shoot is arised, and this is the radicle where the root will be arise in the dicot. And in monocot, what is the main difference between dicot and monocot? Dicot consists of two cotyledons, whereas monocot consists of single cotyledon. I have told you already, instead of cotyledons in monocot, yes, in monocot it is called as scutellum. Okay, in monocots it is called as scutellum. Scutellum is nothing but what? A single cotyledon. Okay, instead of plumule and radicle, here the plumule structure where the shoot is formed called as coleoptile, and the root portion is called as coleorhiza. Understood? So this is what the difference between our dicot embryo and monocot embryo. So this is the axis actually. Below this axis is called as hypocotyle. Above this axis we call it as epicotyle. Epi means above, hypo means below. Okay, so this is the difference between dicot embryo and monocot embryo. Okay, so next, the seed. From where the seed is formed? From the structure called ovule. So hence the seed is also called as fertilized ovule. And these seeds are of two types. Sometimes it is called as non-albuminous and sometimes it is called as albuminous seeds. Okay. And next thing about the seeds are, sometimes within the seeds we know ovule give rise to seeds. Okay, and within the ovule we have studied one structure called as nucellus. Do you remember? The one which gives the nourishment for the developing embryo sac. If the nucellus will persist after the development of seed, if it persists within the seed, then the structure of seed we call it as perisperm. Okay, and for this perisperm there is an example for perisperm. Okay, that is our pepper. Okay, so pepper is the example for perisperm. And seed dormancy, already we know, seed will be kept in the pause stage, dormant stage, by the two phenomenon. It may be outer environmental condition or the internal growth regulators, means plant hormones can also lead to the seed dormancy. That is our ABA, abscisic acid. Okay, so this is what we call it as seed dormancy. So next, seed formation will study, that is here endospermic and non-endospermic is also called as albuminous and non-albuminous, means the seed with endosperm, the seed without endosperm it means. Okay, so we'll study the seed formation. So the zygote grows repeatedly by mitosis. We know how the embryo is formed, that is globular heart-shaped embryo in dicot especially. So how these embryos are formed by repeated mitotic division. So the formed embryo consists of the plumule, that is future shoot, and the radicle, the future root, and the cotyledons. Okay, this is our normal seed formation. Cotyledons plays a role now. It stores food which is needed for the future seed germination. Okay, so next we'll study one by one. Endospermic seed formation. Endospermic related to the endosperm structure. Okay, so we'll see what happens here. The endosperm nucleus, which is 3N, divides repeatedly to form the endosperm in endospermic seeds. This endosperm acts as a food storage for the developing seed. So what happens? Not all the seed contains endosperm. Okay, if percentage wise, if I want to tell you, then 100 within the 100%, 80% of seeds are without endosperm. Okay, and only 20% of seed, after the seed formation also, endosperm is present within that. Okay, so why the endosperms are produced there in such endospermic seeds? To give the nourishment for the developing seed, the endosperm will get persisted. Like example, maize. Okay, in maize or corn, what we eat, if you just look practically the maize seed, the upper yellow part, which is very bulky, juicy, what we like to eat, that part is nothing but endosperm. When you squeeze it, an oval shaped structure you get. That structure is nothing but embryo. Okay, means the seed still persist or they contain endosperm. Such seeds are called as endospermic seeds. Okay, example, maize, remember. So next are non-endospermic, means the seed which doesn't contain any endosperm. Okay, so what happens in non-endospermic seeds? The endosperm is used up in the early stages of seed development, so the food is stored in the cotyledons. So what happens in non-endospermic? Why there is no endosperm in the seed? Because of during the development of seed or the embryo, what happens? The embryo or the seed consumed all the endosperm, so that at the end of the development, we can't see any endosperm. So in the future, what happens? Those two cotyledons only will give the nourishment for the seed. Okay, example, all beans, like groundnuts also. See, if you open a groundnut seed, you will get only two cotyledons. You can't see any endosperm. But when you squeeze out maize, you can see that yellow portion, that is called as endosperm. Okay, so seed formation already we have studied. So this is the structure of ovule. Okay, structure of ovule. The white cells are nothing but the endosperm. What it does, what it gives nourishment for the developing embryo. Next, the green part is embryo, which in turn gives rise to plumule, radicle, and cotyledons. In monocots, it is single. In dicot, it is two cotyledon. What integuments becomes now? Okay, we know the fate of embryo and endosperm. What is the fate or future of integuments? Integuments becomes the seed coats. So you have observed, when you observe a seed, there will be a covering, right? Seed coat. Those seed coat are nothing but our integuments. Okay. Yes. So we know already studied now non-endospermic seed formation. If all the endosperm is absorbed by the developing embryo, the seed is a non-endospermic seed. Already we have studied. See here in the diagram, there is no white portion, white cells. White cells indicate what? Endosperm here. All are here what? Embryo cells. So next, endospermic seed. So if all the endosperm is not absorbed by the developing embryo, the seed is an endospermic seed. Example, maize. Here you can see the white cells as well as green. White cells indicates endosperm, green cells indicates. That is just a diagrammatic representation. Okay, don't depend on these diagrams. These diagrams are only for your understanding. Yes, seed types and structures we'll study further. So what happens? Seeds. Okay, all seeds contains only embryo, which contains plumule, radicle and cotyledon. Some seeds along with this embryo, they contain endosperm for food storage. Okay, this is the difference between endospermic or non-endospermic. Sometimes it is also called as albuminous or non-albuminous. See, this is the structure of endospermic seed. Example, maize. This is a maize seed where you can see a yellow portion. This is endosperm, the seed coat. Okay, cotyledon, that is a scutellum, remember. Okay, plumule and radicle. This is the simple structure of endospermic seed. We'll see now what is the non-endospermic seed looks like. Okay, seed coat, as I told, integuments will form the seed coats. And one more interesting thing, they ask what is testa and tegmen. In the front of seed coat, within the bracket, I have written testa and tegmen. Okay, so while drawing the structure of ovule, we have drawn two integument layers, outer integument layer, inner integument layer, right? So what happens? Both the integuments will form the seed coats, outer seed coat, inner seed coat. So now how to remember which one is outer and inner within this testa and tegmen? I'll tell you one trick, simple trick to remember which one is the outer and inner. Okay, so remember, inner wall or inner layer, inner contains N. Okay, within these two, which word contain N? Tegmen. Tegmen contain N. It means tegmen is the inner layer of the seed coat, inner seed coat. Testa is the outer. Outer, within the outer word T is there, with in the inner word N is there. I have taken this N and T and compared there. Okay, you just remembered either one of these layers, you can remember the another layer. Okay, you remember only inner integument, inner contains N. Within the testa and stegma, tegmen contain N. So tegmen is the inner seed coat, testa is the outer seed coat. Like this you remember. Then what it contains? Simple. Others are same thing, cotyledons, plumule and radicle, but here you can't see any endosperm. So this is what the difference between endospermic and non-endospermic seed. So this is for your simple comparison. You can see here a simple comparison. All other structures are same. They contain the seed coat, they contain the cotyledon, they contain plumule and radicle. The difference is presence and absence of endosperm. Okay, so dicot embryos develop two cotyledons, monocot embryos develop single cotyledons. This is already we know. So you can see their upper common bean structure. Okay, so where there is two cotyledons and below corn you can see there is single cotyledon. So this is what the major difference between monocot plants and dicot plants. How they got the name dicot and monocot you got to know in angiosperms, because of the presence of number of cotyledons. If it is 2, then it is dicot. If it is single, it is monocot. Okay, next, when you see the seedling, along with the integument or a nucellus or embryo sac, there are different structures also. In ovule we have drawn. Okay, the one opening where our pollen tube get entered, which is that opening? Micropyle. Okay, hilum structure where this funicle or the ovule get attached to the stalk, that is hilum. Okay, so in this diagram, this is a structure, simple kidney beans, structure of kidney beans, where you can see the micropyle, okay, hilum, or sometimes it is also called as scar. Okay, then a radicle. So from this radicle, the root emerges. Understood? So this is what the seedling structure. So this we know already, the different parts of seeds and its function. Okay, radicle will be the root, future root. Plumule will be the future shoots. Okay. So cotyledon, this is a leaf of the embryo which can be a food reserve in non-endospermic, or in endospermic they helps in the germination, cotyledons. Testa, and here I have not given tegmen. So remember, testa and tegmen, these are the two seed coats from the two integuments. Okay. So next is the last portion, that is fruit formation. We know from the ovule seeds are formed, and from the ovary the fruits are formed. So here you can see I have given a comparison of a flower and a fruit here. So just see the structure, from which structure which future structures are formed. Okay, so from ovule, can you see here, seeds are formed. From ovary, complete fruit. And this in apple, what happens actually, from the other floral parts the fruits are formed here. Okay, I'll tell you in next slide what are the different kinds of fruits, but just imagine, from the ovary what happens, the ovary becomes the fruit. In all 90% of angiosperms, ovary will form the fruit. Okay, yes. So what is fruit? A fruit is a mature ovary that may contain seeds. Why I have written there that may contain seeds? I have explained you about parthenocarpy in the first chapter. Okay, parthenocarpy, the formation of fruit without seeds, I have told you. So hence I have said there, a fruit may be contains seeds or may not be also. Okay, the process of fruit formation here is stimulated by, with the help of some growth factors. Growth regulators are nothing but the hormones which is present in plants. Those are auxins, gibberellins, cytokinins. You have studied already 5 different hormones. So the process of fruit formation is also helped by the plant hormones which is present within themselves. Okay, yes. Seedless fruits. So this is a beautiful concept, how the seedless fruits are formed. The seeds which is containing the fruits which is containing seeds are the normal concept. Okay, normal phenomenon or behavior of a fruit or of a plant. But seedless fruit formation is a bit diversified concept from the within the angiosperms. So this seedless fruit formation can be of two types. One is genetically, another is by hormones. Okay, genetically what happens, either naturally or by special breeding programs we can perform this seedless fruit formation. Either naturally, like when they undergo parthenocarpy, naturally they don't contain, the fruits doesn't contain any seeds, or genetically, like if we remove or if we manipulate the genes which helps to form the seeds, then what happens, the seeds are not formed. Okay, so examples genetically performed is seedless oranges. Now I have given only one here. Even nowadays seedless guava fruit is also coming, seedless watermelons also coming. Okay, where you cannot see seeds only, means sometimes it happens naturally by the by the performance of parthenocarpy, or genetically they are breeded. Okay, modified. So next factor is our growth regulators or growth hormones, especially auxins. If large amount of growth factors are sprayed on the flowers, fruit may form without fertilization. So what happens? By spraying the large amount of growth regulators, here examples they had given auxins, sometimes gibberellins also. If we want to increase the size of fruit, simply spraying the gibberellins helps to increase in the size of a fruit. Okay, so seedless grapes are one of the example, because we get grapes with seeds also. Okay, some seedless grapes are formed by using this growth regulators. Okay, how do seedless fruits form other than these two, that is genetically and auxin, even the gibberellin also. Okay, we can read these points. Maybe a genetic variety and occur or by natural occurrence. Example, some bananas and cucumbers, you cannot see seeds, right? Spray flowers with auxins or gibberellins, that is growth factors, where the fruits are formed without fertilization. That is parthenocarpy happens when we spray auxins and gibberellins. Gibberellins may also cause bigger fruit to form. I told you already. Next, what happens? We know what is the function of ethylene. Yes, ripening of fruits. Okay, to ripen the fruits artificially, fruits will form ethylene naturally. Okay, but it will take time to form the ethylene within the fruits to get ripe. But what happens sometimes? We spray ethane also, right? I have told you in the previous standards, plant growth and development chapter, I have told you about last growth factor, that is about ethylene. They plays an important role in the fruit ripening process. Okay, yes. So fruits, types of fruits, we know already, we are well acquainted with the fruits. We need fruits one or the other day, we consume the fruits. Okay, some fruits are fleshy, yes, some fruits are dry. Okay, both we consume. What makes fruit fleshy? A fruit consists of that ovarian wall will give rise to a fruit wall called as pericarp. This pericarp consists of three layers, that is inner, outer and middle. Okay, epicarp, endocarp and mesocarp. And mesocarp is the one layer where it is a fleshy and we consume it, we eat it. Okay, so remember fruit wall, that is pericarp. Next classification is false fruits and true fruits. Like as we have come across with the diagram, the first diagram during the fruit formation slide, what we have seen there, a flower with a apple cut end portion of the of that diagram. Okay, so what happens in some, actually the fruits, how they are formed? By the ovary. Okay, if the fruits which is formed by the ovary, if the fruits which is formed by the ovary, then such fruits are called as true fruits. Okay, if the fruits are formed from thalamus, okay, and other structure of the flowers like petals, sepals other than ovary, then such fruits are called as false fruits. The example for false fruits are apple. For true fruits, you can say mango, you can give example. Okay, so now you understood the difference between true fruits and false fruits. True fruits are the one which is formed by the ovary itself. False fruits are the one which is formed typically from the thalamus and other parts of the flowers. Next, parthenocarpic fruits. Already have told, fruits are developed without fertilization, means without seeds. Okay, yes. So next one, apomixis. So don't get confused now. So they may ask the difference between parthenocarpic fruit and apomixis. Okay, or parthenocarpy definition and apomixis definition. Apomixis definition is seed development without fertilization, is referred to as apomixis. Okay, if fruit development without fertilization, then it is called as parthenocarpy. Remember, okay, there is a difference. If there is a seed development without fertilization, called as apomixis. Fruit development without fertilization, called as parthenocarpy. There is a difference. Okay, so try to remember this difference. Okay, the next point is, here what happens, the seed how it directly formed into fruits. No, not sorry, fruits. So seeds, how it is formed. So here the seeds are formed directly by the diploid egg, which forms into embryo, and seeds are formed. So there is no fusion. So next, there is a concept called polyembryony. Means sometimes what happens, we know a single ovule produces a single embryo. Okay, so in sometimes what happens, there is formation of many embryos within the ovule. That condition is called as polyembryony. Okay, how this polyembryony is formed? Sometimes the new cellus, okay, new cellus cells. New cellus understood, the one which gives nourishment, parenchymatous cells. What happens? All the cells, they protrude into the embryo sac, and they develop into many embryos, which in turn we call it as polyembryony. Okay, formation of many embryos, we call it as polyembryony. As the name itself indicates, poly means many, embryony relates embryo formation. Okay, so this is about a few things about polyembryony. So it is the phenomenon of formation of more than one embryo during the development of seed. I have told you already. Okay, so who discovered this polyembryony concept? Leeuwenhoek in 1790, he discovered the polyembryony in the citrus plant like oranges, lemons. This indicates citrus plants. Okay, based on from where they formed, how they formed, the polyembryony is of 3 types. Okay, so the first one is simple polyembryony. In this type, what happens, a number of embryos develops as a result of fertilization of several archegonial cells. Okay, archegonial cells are the one which is present within the ovule. Okay, where it forms many megaspores, right? Remember archegonial cells. So in simple polyembryony, what happens, those many embryos are formed by the archegonial cells. We call it as simple polyembryony. Next is cleavage polyembryony. Okay, in this case, what happens, a single fertilized egg give rise to number of embryos. Okay, after fertilization, the egg fertilized into like zygote is formed. That fertilized egg, what happens, they give rise to number of embryos. And the last, adventitive, or it is also called as rosette. Okay, here what happens, some structures will be there within the embryo. Those structures will give rise to the different types of embryos. Okay, so these are the 3 different types of polyembryony. Simple means which is formed by archegonial cells. Cleavage means which is the embryos are formed by fertilized egg, single fertilized egg. Adventitive means they are formed by the additional cells. Okay, thank you. So next we'll start with the new chapter. Okay, so I'll decide whether to start with the human reproduction or the different unit, because even the YouTubes from the PU board also they are uploading. So go through the human reproduction videos from the PU board. Soon I'll upload the new video. Okay, thank you.